Okay. All right. So we're very excited to have Dr. Peter Thompson again speaking with us. He's had some great talks on at the Sugar Science in the past, and this time he's also accompanied by Jasmine Pipella, a PhD student in the Thompson Laboratory. The title of the talk is Old Dogs, New Tricks, T Cells Drive Beta Cell Senescence in Type 1 Diabetes, and a short bio. Dr. Thompson completed his PhD at the University of British Columbia in 2016. He carried out postdoctoral training at the UCSF Diabetes Center, where his work discovered beta cell senescence as a contributor to type 1 diabetes. In 2020, he started his independent research program in the Department of Physiology and Pathophysiology at University of Manitoba, Canada, at the Children's Hospital Research Institute as a member of the Pediatric Diabetes Research Theme DREAM. His lab investigates islet cell biology and stress responses and their interactions with the immune system in type 1 diabetes with the goal of developing new early stage therapies. Jasmine Pipella completed her Bachelor of Science with honors with a major in biochemistry and a minor in psychology at the University of Jamestown in 2021. And she's currently a PhD student in Dr. Peter Thompson's lab in the Department of Physiology and Pathophysiology at University of Manitoba and the Children's Hospital Research Institute of Manitoba, and her research focuses on studying the role of the immune system in beta cell stress pathways. Thank you both so much for joining us. Really interested to hear this talk, and I'm dropping into the chat that we'll take questions at the end and then a link to the newest paper. So yeah, thanks for joining.
Fantastic. Thanks, Monica, for that warm welcome and introduction. Thanks to everyone who can join us today. So we're really excited to share some new work out of the lab, old dogs, new tricks, kind of a spin on that phrase, can you teach an old dog a new trick? And hopefully today we'll be able to convince you that T cells have been doing something that we wouldn't have anticipated in the context of type 1 diabetes, influencing and driving really the development of beta cell stress pathway known as senescence. And this is really outstanding work done, led by my PhD student, Jasmine. So I'm going to give a little bit of a brief background and then going to hand it over to her to tell you the rest of the study. Right, so we'll get started on the first slide there. So I just wanted to, start this discussion with reminding you all, and maybe if you're not familiar, that the classical autoimmune paradigm for type 1 diabetes has been challenged and expanded in recent years. Classically, we considered that this was really just an autoimmune-driven disease where T cells simply selectively target the pancreatic beta cells, and that was the end of the story. But I would say in the last 10 or so years, it's become increasingly clear that there's a very complex crosstalk going on between the islets and the immune system in type 1 diabetes. And there's things that are happening at the level of the beta cell, notably various different stress pathways that are also contributing to the development of type 1. And you see some of them here. There are lethal responses that beta cells can undergo. We consider this to be things like the terminal unfolded protein response, or a UPR, where beta cells experience unmitigated ER stress and then they'll die. There's evidence for other forms of stress, things like adaptive EPR and autophagy. Work by Amelia Linneman's group has demonstrated autophagy in the context of type 1. And then work that we've been really pushing forward has discovered that some beta cells acquire senescence during type 1. And so that's really been the focus of my research program and some of the work that we're going to be talking about today, understanding senescence and where it comes from. So just as a basic primer, for those who aren't familiar, senescence is classically a tumor suppressive mechanism. And the idea is that the cell will experience different forms of damage that can lead to the acquisition of a cell cycle arrest. And this cell cycle arrest is considered to be permanent or stable. And the cell undergoes a variety of molecular changes downstream of that, things like activation of a DNA damage response, upregulation of cyclin-dependent kinase inhibitors, which are enforcing the cell cycle arrest, changes in the lysosomal pathway, so the elevation of the senescence-associated beta-gal activity, Notably, senescent cells are not cells that are en route to death. This is a really important concept, particularly for the work that we're going to describe. But just in the field, it's important to note that senescent cells are persistent. They have upregulated their pro-survival pathways. And most notably among those pathways are the BCL2 family of anti-apoptotic proteins. So this confers apoptosis resistance. And then these cells they can become highly inflammatory through the secretion of what's known as the senescence-associated secretory phenotype, or SASP. And this consists of a collection of cytokines and proteases and shed ligands and receptors that are highly immunogenic. And so these cells can arise in various forms of stress, but it's important to point out also that they have important physiological roles. So senescence is obviously around for a reason. It serves a purpose in the organism. And you can see some of those functions here, things like wound healing, tissue regeneration, and so on. However, one of the sort of emerging paradigms that's come out of the senescence literature is that while senescence on its own isn't necessarily a bad thing, if the immune system can recognize these cells and respond appropriately, these cells can be cleared to promote tissue remodeling and regeneration. However, when this process fails, senescent cells can accumulate, and this leads to a deleterious outcome for the tissue, for the organism, things like chronic inflammation and so on. So what is the evidence that senescent beta cells play a role in type 1 diabetes? Well, this is going back to some work that we did while I was a postdoc at UCSF with Dr. Anil Bhushan, where I uncovered the senescence signature in a population of beta cells in the NOD mouse model. Most of you are familiar with that model, spontaneous autoimmune diabetes model. These senescent beta cells arise, pre-diabetes in these mice. So before the onset of hyperglycemia, you have a subset of beta cells that are becoming senescent based on a variety of these molecular signatures that we identified. And what we found to really probe the functional relevance of these cells, we used a small molecule inhibitor of the BCL2 family protein, particularly BCL2 itself, known as ABT199. And there's a variety of agents in this family of compounds that can target the BCL2 family and have been used as senolytics in other contexts. So we used ABT199 to show that if you administer this treatment intermittently in the late pre-diabetic stage to NOD mice, you can maintain their euglycemia so they don't progress to develop type 1 diabetes. And notably, we probed the immune compartment to see what role the senolytic might have on immune cells. And we found no evidence to support that senolytics have any direct effect on any of the major immune cell populations involved in type 1 in this mouse model. So this really is a strong evidence that senescent beta cells contribute to the disease. And I would also point out here work in parallel by Cristina Aguayo-Mazzucato's group and Susan Bonneweir at Joslin showed a similar effect of senescent beta cells in accelerating type 2 diabetes in type 2 models. So we think that senescent beta cells are not just unique to type 1, but they are occurring in these different contexts where beta cells are accumulating stress. But we had this big question, and it really became the focus of my lab for the first few years, really trying to understand what causes beta cells to become senescent. And you can think about this from two perspectives. On the one hand, there's things that are happening inside the beta cell, so intrinsic mechanisms that might predispose a beta cell to undergo a senescence response to different forms of stress. But we also wanted to interrogate and think about questions around, what are the extrinsic factors that are impinging on beta cells that lead them to become senescent? And so in the context of type 1 diabetes, as you know, we have this very helpful, clinically relevant staging model where you go from stage 1 with the presence of 2 or more autoantibodies to stage 2 with dysglycemia to stage 3 with the onset of hyperglycemia. And studies we did in using donor pancreas tissue sections from NPOD supported that senescent beta cells begin to accumulate even as early as stage 1 and 2, again, before the onset of hyperglycemia. And we could mark these beta cells by the presence of a variety of different molecular signatures. So again, P21, P16, DNA damage response, the SASP, markers for the SASP, as well as upregulation of BCL2 family proteins. But we wanted to really kind of address this thought of, I told you earlier that senescent cells really only accumulate when there's sufficient stress to cause them to become senescent, and there may be an impairment in the mechanisms that lead to their removal. So in this study, we really wanted to think about the question of what's happening at the level of senescent beta cell generation and what role the immune system plays specifically. And so that really leads into this work that we did, this study that we did to specifically interrogate the role of T cells in beta cell senescence. All right, so I'm going to turn it over to Jasmine, and she's going to lead you through the rest of the talk and describe the study that she led. Jasmine, if you will.
Thanks, Peter. So yeah, I'll just start by telling you a little bit about the first question we had. So we really wanted to address the question of what role does the autoimmune process play in beta cell senescence during type 1 diabetes. And so to do this, we used immunohistochemistry for established senescence markers in well-characterized genetically immune deficient NOD mouse models that had their adaptive immunity affected. And so we used both the NSG and NOD-REG1 knockout mouse strains, which both don't develop diabetes due to the lack of mature T and B cells. And we also looked at the NOR mouse strain, which is resistant to insulitis, so they don't develop type 1 either. And what we found using IHC is that the beta cells expressing the cyclin-dependent kinase inhibitor P21 and SASP marker MMP2 were substantially reduced in all of the mouse strains where their adaptive immunity was affected compared to wild type controls. And it's important to note that we did do this at the pre-diabetic stage. So all of the mice were between 12 to 14 weeks. So I just really wanted to emphasize that this data was able to suggest that an intact autoimmune destruction process is required for the accumulation of senescent beta cells in the NOD mouse model. But then we wanted to directly test whether the autoimmune process itself could drive beta cell senescence. So to do this, we performed an adoptive transfer experiment where we transferred splenocytes from newly diabetic NOD female mice, and we transferred them into 8-week-old immune deficient NSG mice. And as controls, we also had a separate group of NSG mice that received PBS only. And so following the transfer, we monitored their glucose for about 3 to 4 weeks. And when the first recipient mouse became diabetic, that is when we sacrificed the whole cohort and we collected both pancreas and spleen tissue for further analysis. And I'm not going to show it here, but we did do flow cytometry on the spleens, and this confirmed that there was repopulation of the CD3 T cells in the mice that received the splenocyte transfer compared to the PBS controls. But most significantly, when we did IHC, we showed that there was a significant increase in beta cells that were positive for both P21 and MMP2, which are two of our main markers for senescence. And it was also important to show that the insulitis scoring from these mice, the mice that received the immune cells had more peri-insulitis and insulitis, showing that the immune cells were actually able to migrate to the pancreas. And so these results just demonstrate that the autoimmune destructive process is both necessary and sufficient to drive beta cell senescence during type 1 diabetes development. And so far we've seen that when we remove the autoimmune system, beta cell senescence is markedly reduced. And when we reintroduce the immune cells through adoptive transfer, the senescent beta cell phenotype comes back. And so this tells us that the autoimmune process is both necessary and sufficient to drive beta cell senescence, but it raises an important question in which what immune cells are actually responsible for this effect. And so given that T cells are the primary drivers of autoimmunity in type 1 diabetes, this brought me to my main research question of what role do T cells play in beta cell senescence during type 1? And so many of you have probably heard or are well aware of teplizumab, which is an anti-CD3 monoclonal antibody. And it works by targeting CD3, the CD3 molecule on T cells, and it essentially dials down the autoimmune attack against pancreatic beta cells. And by doing this, it can help preserve insulin producing beta cell function. And so I'm just going to go through a little bit of the history in the NOD mouse models. So as Peter mentioned, the NOD mouse model is a common model for spontaneous type 1 diabetes, and anti-CD3 showed very strong results in this model. So when a single dose was given at stage 1, it was actually able to prevent type 1 diabetes in these mice. And more commonly studied when given for 5 consecutive days after diabetes onset, anti-CD3 was also able to actually reverse the disease in the NOD mouse model. Yet these results were not completely recapitulated in clinical trials. And so in people, teplizumab is often given as a 14-day infusion, and the clinical trials have been able to show that it can delay the onset of symptomatic type 1 diabetes in some high-risk individuals, and also slow the decline of beta cell function in some new onset type 1 diabetes. And based on these findings, the FDA approved teplizumab in November 2022. And so building on what we know from previous studies in NOD mice, we really wanted to look at what happens if you give the treatment intermittently, especially during the late pre-diabetic stage when senescent beta cells are actually starting to accumulate. And so to study the role of T cells in this pre-diabetic phase, we designed a new treatment model. And so instead of that traditional 5-day regimen, we treated the female NOD mice once per week from weeks 9 to 13 with IP injections of anti-CD3 antibody. And as a control, we use species-matched IgG. And so this really allowed us to ask whether intermittent dosing could actually modulate the immune response at this critical stage of the disease. And we were able to validate this treatment using flow cytometry. So after 4 weeks of treatment, when we euthanized the mice, we saw that the total CD3 T cells remained partially depleted in the mice that received anti-CD3. But when we looked at antigen-specific responses, we found a significant decrease in insulin-reactive CD4 T cells, but not in IGRP-reactive CD8 T cells. And similarly, we saw a strong reduction in overall CD4 T cells, while CD8 T cells were largely unaffected. But this specific effect of this intermittent regimen that we see fits with previous work showing that CD4 T cells are more sensitive to anti-CD3 in the NOD mouse model because they express higher levels of CD3 compared to CD8 T cells. But unlike earlier studies, our model did not expand the regulatory T cell population. So the main effect here really was on the autoreactive CD4 T cells. And so the key takeaway from this slide is just that the intermittent CD3 antibody treatment selectively reduces autoreactive CD4 T cells in the NOD mouse model without broadly depleting all T cell subsets. And this really provided us a model to investigate how T cell modulation could influence beta cell senescence and disease progression before the onset of overt diabetes in these mice. And so we next wanted to look at how this intermittent CD3 treatment could actually translate into better disease outcomes for these mice. And so first we saw that the treated mice had a significantly larger insulin positive stained area. And we also found significantly less insulitis as expected. So you can see that the mice that received CD3 had way more intact islets and fewer islets that were heavily infiltrated with immune cells. And functionally, this translated into improved glucose tolerance. Although this effect was mild, we did see a reduction in area under the curve in the mice that received the anti-CD3. And so overall, this intermittent CD3 antibody, we were able to show that it slows disease progression in the mouse model. But the next thing we wanted to ask was, what about senescence and whether this intermittent anti-CD3 antibody treatment could influence changes in senescence. And so on the left, you can see that there's immunostaining for both P21 as well as MMP2. And with both markers, we saw a significant reduction in beta cells that were positive for either P21 or MMP2. And so we wanted to confirm this at the gene expression level. And so in islets that were isolated from treated mice, we were able to show that there was a reduction in the gene CDKN1A, which encodes P21, as well as the SASP genes MMP2 and IL-6. And so overall, this treatment lowered both protein and mRNA markers of beta cell senescence. But we really wanted to look at other pathways to see if they were also being affected. And so when we looked at the unfolded protein response marker spliced XBP1, really surprisingly, we saw no changes between the two groups. And importantly, we also saw no changes in the number of alpha cells, in proliferation marker Ki67, or beta identity markers like PDX1. And so interestingly, when we stratified senescent beta cells by insulitis stage, we also found that in the control group, P21 positive cells were enriched in islets with insulitis, but this relationship largely disappeared in the mice that received anti-CD3. So this experiment really showed that there was a link between immune infiltration and senescence. And I'm not showing the data, but it's in the paper if you're interested. But we also asked the question of whether CD4 and CD8 T cells can contribute to beta cell senescence individually. So we selectively depleted each population in the NOD mouse model. And then we found that both insulitis and senescent beta cells were significantly lowered in both experiments, indicating that both subsets are required for the process of beta cell accumulation. And so taken together, these results really show that the intermittent CD3 antibody treatment reduces beta cell senescence without disrupting other stress pathways, such as unfolded protein response. And so since the anti-CD3 treatment limits beta cell senescence through immune modulation, and like Peter mentioned earlier, senolytics like ABT199 can directly target senescent beta cells and delay progression to type 1 in the NOD mouse model. We really wanted to ask the question of whether we could combine these two approaches to have additive benefits. And so again, we treated female NOD mice at the pre-diabetic stage, so once per week for 4 weeks with either IgG or anti-CD3. And then starting at 10 weeks, the mice also received either ABT199 or vehicle. And so the mice that received the combination of both ABT199 and anti-CD3 showed the greatest reduction in insulitis, as well as the best glucose tolerance. But interestingly, we didn't see any changes in overall insulin positive stained area between any of the three groups, suggesting that maybe beta cell preservation wasn't really the main driver here. But when we moved on to look at senescence, both ABT199 plus IgG and ABT199 plus CD3 did show a reduction in P21 and MMP2 positive beta cells, but the effect was definitely strongest in the mice that received the combination therapy. And it's also important to note that we didn't see differences in unfolded protein response marker spliced XBP1 in any of the three groups either. And so the key takeaway here is really that the anti-CD3 and senolytic treatment can act through distinct but complementary mechanisms. And so when they're combined, they have additive effects in slowing disease progression and reducing beta cell senescence in the NOD mouse model. And so up until this point, I've kind of just shown you data in the NOD mouse model, but we also wanted to address human relevance here. And so although the gold standard experiment would have been to co-culture autoreactive T cells with human islets from the same HLA haplotype, and then that would have allowed us to directly observe how T cells trigger beta cell senescence in their native context. Practically, that's really difficult to do because the immunology is very complex. So instead, we used a different approach because we know that in type 1 diabetes, activated helper and cytotoxic T cells can produce high levels of interferon gamma and tumor necrosis factor alpha. And together with IL-1 beta from macrophages, these cytokines can create a highly inflammatory islet microenvironment. And this cytokine mix has commonly been used to study beta cell stress and beta cell death, but we were unsure if these inflammatory signals from autoreactive T cells would be enough to drive beta cell senescence in human islets. And so to test this, we treated human islets with the cytokine mix that I mentioned with interferon gamma, TNF alpha, and IL-1 beta. And we did this for 48 hours. And then we did a drug washout on the islets. And we did this because we wanted to see if the effects would be able to persist after the drug was actually gone. And as a positive control, we used bleomycin, which is like our well-established DNA damage driven senescence positive control. And what we found was that the cytokine treatment was able to induce a senescence program that was similar to bleomycin in human islets. So you can see here that the cytokines were able to upregulate CDKN1A, which encodes P21, and that's similar to bleomycin. But if you look at CDKN2A, which encodes P16, only the cytokines had an upregulation. So this may indicate that unlike bleomycin, cytokines are able to increase P16. So they might be acting through a slightly different senescence program. And then looking at some of the SAS genes, we didn't see any upregulation of GDF15 with the cytokine mix, but we did see it with for CFCL8. And then importantly, neither the cytokines or bleomycin activated the UPR gene CHOP, which was consistent with previous work that has been done. And so the data isn't shown here again, but if you're interested, we wanted to further confirm these findings at the protein level. So we did Western blots and showed that the cytokines are able to increase P21 in human islets. And we also used the human beta cell line endo-C beta H5 cells. And with those cells, we were able to show that there is a stable senescence-like phenotype after cytokine treatment at both the gene and protein level. And so the main point from this set of experiments is really that type 1 diabetes related inflammatory cytokines can recapitulate some of the stable phenotypes that we see during senescence in human islets. And so to wrap things up, our work really focused on how autoreactive T cells and cytokines how autoreactive T cells and the cytokines that they release can contribute to beta cell senescence during type 1 diabetes. And when you deplete those autoreactive T cells, you can actually reduce the accumulation of beta cell senescence. And so in the NOD mouse model, we showed that the intermittent anti-CD3 treatment reduces T cells, which in turn decreases beta cell senescence, lowers insulitis, and improves glucose tolerance. And turning to human islets, we also found that a mix of type 1 diabetes related cytokines is sufficient to drive a stable senescence program. And this was really reflected by the increased expression of both senescence and SAS genes, as well as senescent proteins, even after cytokine washout. And so one of the most interesting findings from our work is really that when we deplete the autoreactive T cells with anti-CD3, it specifically reduces beta cell senescence. But importantly, these don't alter the UPR-driven stress pathways, showing that senescence is a distinct response and distinct phenotype. And so from a clinical standpoint, this work really offers two kind of important insights. And so first, it's important to take away from this paper that autoreactive T cells don't just kill all beta cells, and that they have the ability to push healthy beta cells into stressed states such as senescence, where these cells they stop working properly and their function starts to decline. And second, because anti-CD3 and senolytics work through distinct pathways, combining them could provide additive benefits in slowing or preventing type 1 diabetes progression in some of the individuals who are at risk for developing type 1. And so to close, I just want to thank Dr. Thompson, as well as my lab members and committee members. And I'd like to acknowledge all of the funding that we had that supported this project, as well as both the Integrated Islet Distribution Program and ADI and all the donors and their families. And thank you to the Sugar Science Think Tank for inviting us to share our recent work. And if you're interested in reading more, the paper is here. So we're happy to take any questions you might have.
Thank you so much, Jasmine and Peter. I'll open it up to questions. If you have a question, if you'd like to raise your hand or drop something in the chat, we can read it. Here's something already. Here's from Todd Briscoe, University of Florida. Is there an impact of senescence drug on tissue-resident APCs and islets? IOW, could this be an indirect impact? As a follow-on, does T-cell exhaustion induced by anti-CD3 mimic this impact?
Do you want me to answer?
Yeah,
Peter,
go ahead.
do you want Okay. I mean, I don't know if there's for sure a lot out there on tissue-resident antigen-presenting cells, but some of the work that I'm doing now is looking at different immune cells that could be involved in this, as well as the tissue-resident ones. I think it's just pretty difficult to get enough of the tissue-resident immune cells to do things like co-culture assays. I don't know if you have anything to add, Peter.
Yeah, I would say, it's something we haven't looked into. We are not really, we haven't really thought a whole lot specifically about, how different, ways that senolytics might have more subtle effects on different immune cell populations, especially those that are residing in the islet, things like macrophages or dendritic cells. But yeah, that's definitely something we want to look into a little bit further. And then the follow-on around T-cell exhaustion, again, we haven't done those experiments. It would be really interesting to look at that, though.
We got some accolades there in the chat. Very nice talk. Ramuruchi Jane, did you check the effect of ABT 199 on GSIS in the cytokine-treated model of beta cell line?
No, we only did GSIS on the mice that were only treated with either IgG or CD3, so nothing to do with the senolytic. And we also didn't do GSIS on the human islets that were treated with cytokine. So.
Okay. This is something we spoke about offline prior to the talk, and here it is again from Cassian Ayoane. Hey, very interesting. Ulf Algren just put out an interesting paper on bioRxiv. Only a fraction of the residual beta cells were located within islet structures. Instead, the absolute majority were present as extra islet beta cells, either as scattered individuals or clusters of beta cells, spatially separated from all other endocrine cell types. How do you think this translate into mouse versus human differences? And just as an aside, the patient they looked at was diagnosed at 50 years and had type 1 for 12 years prior to tissue access by Ulf Algren's lab. They used that specialized 3D imaging to capture these data. So yeah, that's a very interesting question, right?
Yeah, I mean, we don't really have a lot of insight into that at this point. The question of why these beta cells are scattered, are they newly proliferated, or where are they derived from, is really interesting and really important. I think there's a lot of exciting work being done on the crosstalk between beta cells and other endocrine cells, and if you find these spatially isolated beta cells kind of floating around in different regions of the pancreas, it kind of raises that question of, are they functional? Can they respond to glucose? And perhaps, work by Guy Rutter's team and others, Vera Kravitz, looking at heterogeneity in beta cell function, right? These functional subsets like leader and hub cells, if they don't have the right connections, they're not going to be able to function properly. And that might be really interesting piece of the puzzle as to, why we have these beta cells that are still present. They survive the autoimmune attack, but they don't work properly. So yeah,
Yeah.
very interesting point.
You wonder if the zones that retain the beta cells align with like a lower senescence or a higher senescence,
Yeah,
right? Like
exactly.
that's another kind of curious thought. Okay,
And
let's.
if I add, oh, sorry, I was
Oh
just
yeah,
going to
no.
add that it's also difficult to, with human islets, even if you stain like sections from NPOD or something, there's not a lot of immune infiltration. So seeing the same thing between mouse and human, it's hard to make the link that insulitis, the link between insulitis and senescence in human.
Mm-hmm. Just sort of, we have a couple more questions. Just tapping on that. What is the like? How can you detect senescence, or can you detect senescence in humans in stage 1, 2, who are progressors to type 1 diabetes? Like, are there senescent markers in the human that could be detected? Just to kind of elaborate there a little bit.
I mean, there's work going on in our lab right now. One of our master's students was working on a project with circulating biomarkers so some of those SAS factors that I mentioned, like GDF15 or CXCL8, and there's a number of others, can be detected in the samples from human patients. So we've shown a link between that. I think it'll be out soon, hopefully.
Oh, good. I can't wait to see that. Yeah, that would be really, that's going to be really interesting. Dr. Nwasu from New York, would you like to unmute yourself?
Yeah. Can you hear me?
Yes.
Okay. Hi. Peter and Jasmine, great talk. Thank you so much. The way I'm looking at this as a clinician is that you're looking at the extrinsic factors that could lead to beta cell demise, as opposed to looking at the intrinsic factors that determine beta cell health. That could also be combined with your approach to get to 100%, because I have not heard the percentage of protection that your intervention is affording the beta cells. So that's one. Even the story of teplizumab, we know that I heard the beta cell stress mentioned only once during the presentation. Teplizumab, again, is an extrinsic, player. It doesn't do anything within the beta cell because it does not decrease pro-insulin to C peptide ratio, which is a marker of beta cell health. So my question then is there a way for you to get intrinsically to the beta cell? Because autoimmunity is not the whole story. The stories have been going on for more than 25 years, and we haven't gotten closer to the goalpost yet.
Yeah, those are great points. Thank you so much for those questions. So getting to the first point you mentioned around what is the level that's afforded directly intrinsically to the beta cell. So I can really only comment on the studies we've done in the NOD mouse, and kind of going back to the slide that I mentioned around using the senolytic directly on the mouse model to show that you can delay or in some cases prevent diabetes in these animals. So we see about a protection effect of, we get about 75 to 80% diabetes in the control or vehicle injected, and then it goes down to about 25 to 30% in the animals that have been dosed with ABC 199. So in terms of, how effective this intervention is, I would say it's quite effective. Of course, it's not working in every case, and why do some mice that still receive that drug progress is an open question. But the nice thing with ABT199, in the sense of getting at your second point around, again, the intrinsic things that are going on inside the beta cell, is that we're directly targeting a pro-survival pathway in the senescent beta cell with that agent. So, again, the evidence we have for that is that we see upregulation of BCL2 in senescent beta cells in the mouse model. And based on the study that we did, we showed that when you administer the senolytic, it can directly target, just dissociate the interaction of BCL2 with its binding partners, things like BAX and BAD, and trigger intrinsic apoptosis in the senescent beta cells preferentially. So we think that agents like this can be acting at the level of the beta cell, but it's important that you raise that important question of, like, we need to think not just about immune interventions, but things that you can do to target the beta cell. I know that there's a lot of fantastic work coming out on things like polyamine biosynthesis with a paper that came out from Linda DeMaglio and Emily Sims and Regu Mamira, showing that you can inhibit polyamine biosynthesis in the beta cell, and that also has a protective effect in type 1. Of course, there's the work with imatinib from Steve Gittleman's group. So there's a lot of different agents, I think, that are being thought of to be acting at least at some level on the beta cell to modulate the stress responses. Now, in every case, can we rule out that there's not some sort of simultaneous effect on the immune system? I think that's very challenging to do. And potentially, maybe there are combinatorial effects. I think we learned that from the work that we saw from Thomas Kay and the effects of JAK-STAT inhibitors, the really beautiful clinical trial work that came out on type 1 interferon inhibition with JAK-STAT inhibition, and seeing an effect on the beta cell as well as an effect on the immune system. So it might not be necessary to always be able to separate those two effects if we can see and show that it's a safe, beneficial effect in terms of preserving C-peptide, in terms of slowing disease progression. So that's what I would offer in terms of the questions you raised. But yeah, it's a very valid point, very important point.
Thank you, Peter.
Great discussion. Just two more questions in the interest of people's times. Just from City of Hope, Brian Liu, congrats on the great talk and excellent work. If I understood correctly, beta cells can undergo senescence naturally. Could you comment on the differences between T cell induced senescence and natural senescence?
I'll let Jasmine take that one.
Okay, sounds good. Yeah, so there's actually, we wrote like an opinion article on immune surveillance, and we kind of talk about the differences between what we would define as natural senescence versus stress-induced senescence. And there are some like key differences. So from what I know, in natural senescence, some of the phenotypes are slightly different. So we usually see more of an upregulation of P16 and not necessarily the cells don't exhibit SASP or DNA damage or the pro-survival phenotype that we see in something that's more of a stress senescent state, where we see upregulation of cyclin-dependent kinase inhibitor P21, as well as secretion of those SASP factors and DNA damage. But I don't know if Peter has anything to add.
Yeah, no, I think that's exactly it. Is we can distinguish some of these signatures from beta cells that are aged and they have predominantly a P16 phenotype that doesn't seem to involve P21 or DNA damage or SASP, from those that seem to have become senescent through a stress-mediated process, and those ones have the P21, SASP, and DNA damage. But it is an important question, and heterogeneity in senescence is another emerging concept. Our work and that of others, Christina Guaya-Mazzucato as well, has suggested that there are distinct subpopulations of senescent beta cells that may coexist. So, what is it that turns maybe an aged beta cell into like a stressed senescent beta cell? Are these states interchangeable? Are they states versus, subtypes or whatever term we want to use to describe that heterogeneity? That's still something that's really emerging from the field and being worked out. So yeah, I think stay tuned because the next few years are going to be really exciting in this space. And as we kind of have some tools to probe these things further, we're going to be able to hopefully delineate these senescent populations and make better conclusions about their phenotypes and function.
This is a super sort of outlier question, but has anyone studied the pancreatic beta cells in samples from progeria patients in terms of like, is there senescence that occurs in those people that is like a natural senescence that you could have as like some kind of comparative benchmark?
Yeah, I don't think I've seen any work where anyone has studied senescence from, progeroid patients or patients that might have some rare mutations that lead to these progeroid syndromes. That would be really exciting to do. As we all know, working with human tissue specimens is very challenging and obviously a great privilege. So whenever we have the opportunity to do that, agencies like MPOD and networks like that have really led the way in providing those resources to the community. But yeah, it is a challenge and getting those tissues.
Well, maybe something for NPOD to think about as senescence becomes more center stage. It might be something to think about. Okay, very last one. Have you seen any senescence phenotype in the diabetic exocrine, of the non-mice and pod samples. In some pictures, I can see exocrine staining for P21, etc. Patients with T1D develop pancreas atrophy prior to onset. And that's again from Cassian.
I mean, I haven't actually looked at that to quantify anything, but that would be something interesting to look at because we already have this section.
A lot of projects up there in the Thompson labs, exciting times. And you had a lot of accolades, a lot of great commentary in the chat. So congratulations on this paper, and we look forward to the next one coming out regarding some of the topics we discussed. And great to see you both again. And again, it was really, really an excellent conversation. So have a great rest of your day, and we'll see you again soon, we hope.
Awesome.
Thank
Thank you so
you.
much, Monica.
Thanks
Thanks
again.
to everyone.
Thanks, everyone. Bye-bye.